Hello friends, in the second part of Hmp shunt pathway, I will be discussing the oxidative and non-oxidative phase of Hexose MonoPhosphate shunt pathway, and I will be going through the regulation of Hmp pathway also, and I would like you to go through the first module, that is the first part of Hmp pathway, in which we had a discussion on the significance of Hmp pathway. So let's start. So Hmp shunt pathway part second, Hmp. This is hexose monophosphate shunt pathway because hexoses are involved. Pentose phosphate pathway because pentoses are involved, and 6-phosphogluconate oxidative pathway because this particular substrate 6-phosphogluconate is also involved. Now there are three main significances for Hmp pathway, which have been already discussed in the module 1 of this Hmp pathway, part 1. Now, first one is it provides NADPH, the reducing potential. Second one is it provides ribose, the essential precursor for synthesis of nucleotides. And the third one, as illustrated here, glucose giving glyceraldehyde 3-phosphate, then pyruvic acid, this is glycolysis. And the third significance is that the substrates in cellular pool can be shared between glycolysis and Hmp pathway. So glucose and glucose 6-phosphate can be taken to this alternative pathway, namely pentose phosphate pathway, as well as the other substrates. And this process, Hmp process, does not provide ATP. It does not utilize or provide ATP. So that is one point just to be remembered. Then this process occurs in the cytosol. Now let us see further into Hmp pathway. The Hmp pathway is divided into two phases, or it is taking place in two phases. First one is oxidative non-reversible phase, and the other one is non-oxidative reversible phase. So this is the nutshell of oxidative and non-oxidative phases of Hmp pathway. So this is oxidative non-reversible phase, where you can already see NADPH, 1 molecule, 2 molecules, and then non-oxidative pathway, you can see the ribose molecules here. So this is being a reversible phase. Any of the substrates can be used for synthesis of ribose and for the utilization of ribose. So we can see the oxidative phase first of all in detail. After that we will go to see the non-oxidative phase also. The oxidative phase starts with the substrate glucose 6-phosphate, and it's not glucose. Actually glucose gets converted or esterified with the phosphate group to glucose 6-phosphate instantaneously, because glucose will pass the cell membrane of a particular cell, unlike glucose 6-phosphate, which will be kept inside the cell. And the oxidative phase begins with the dehydrogenation of glucose 6-phosphate at the first carbon by an enzyme glucose 6-phosphate dehydrogenase, which catalyzes the transfer of a hydride ion, and that is a hydrogen atom which has captured an extra electron. So a hydride ion from glucose to NADP plus, so as to become NADPH, nicotinamide adenine dinucleotide phosphate reduced form. So this actually releases an H plus ion also, which again is lost from glucose 6-phosphate. And the resultant molecule is an intramolecular ester called 6-phosphogluconolactone here. Now the second step, this is 6-phosphogluconolactone gets converted to 6-phosphogluconate with the help of the enzyme lactonase, which is a hydrolytic process. As you can see here, it involves hydrolysis, and this actually involves the opening of the cyclic structure or the ring structure of glucose here. So this is what happens. As also the molecule gets prepared for the decarboxylation reaction, which has to take place as the next step. Now next step is 6-phosphogluconate gets converted to ribose 5-phosphate in the presence of 6-phosphogluconate dehydrogenase. Let's see how. Here again This is actually oxidative decarboxylation. So it involves an oxidation reduction reaction as also a decarboxylation reaction. The carbon dioxide is lost from the first carbon here, as here it can be seen carbon dioxide is lost, as well a pair of electrons are transferred from 6-phosphogluconate to NADP plus to become NADPH, nicotinamide adenine dinucleotide phosphate reduced form. As well, hydrogen is also taken up by NADP plus so as to become NADPH. So that's what happens, 2 electrons and 1 hydrogen. So that's how it becomes NADPH. Now this is the final step of oxidative phase, which gives rise to the second molecule of NADPH. So 2 molecules of NADPH are with us at present. So this is illustration for the oxidative phase reactions. You can pause and go through it. And what we have now at present with us are NADPH. Again, NADPH, 2 molecules, then 1 molecule of Ribose 5-phosphate, with which we will go to the non-oxidative phase. Non-oxidative phase starts with an isomerization reaction where Ribose 5-phosphate is isomerized to Ribose 5-phosphate with the help of the enzyme phosphopentose isomerase as shown here. Here, Ribose 5-phosphate is actually a ketose, a ketopentose 5-carbon sugar, and Ribose is a naldopentose, and this is obviously having a CH group at the anomeric carbon atom. And next step also involves a type of isomerization reaction, which is actually an epimerization with the help of the enzyme phosphopentose epimerase. Ribose, another molecule of Ribose 5-phosphate, gets converted to cellulose 5-phosphate, as you can see here. So this is epimers. These two are epimers because it differs around a single carbon atom, as can be seen here. This carbon, the third carbon, is HOH in Ribose 5-phosphate, whereas here in cellulose the OH is on the left side. In the next reaction actually involves both these products. This one, Ribose 5-phosphate and cellulose 5-phosphate, and it's a kind of a transferase type of reaction, which involves a transferase enzyme, where, as shown here, the cellulose 5-phosphate and Ribose 5-phosphate, both these products in the first 2 steps, a block of 2 carbons is taken from here and placed on Ribose 5-phosphate so as to form the 7-carbon pseudoheptulose 7-phosphate. So this is a 5-carbon common, another 5-carbon common, total of 10 carbons. It actually becomes a 3-carbon glyceraldehyde 3-phosphate and a 7-carbon pseudoheptulose 7-phosphate. Obviously, transketolase is the enzyme involved, which is a class of transferase enzyme, which transfers a block from here of 2 carbons here to this, so that pseudoheptulose 7-phosphate is formed. Now, in the next step, this involves these 2 products here, that is glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate, which are having a combined strength of 10 carbons now. So what is the next reaction? Yeah, pseudoheptulose, as I said, has pseudoheptulose and glyceraldehyde 3-phosphate with the help of an enzyme, another transferase type of an enzyme, transaldolase, and the products formed are fructose 6-phosphate and erythrose 4-phosphate. Again, 10 carbons here give rise to another combined strength of 10 carbons. And what is the reaction? Yes. Now, pseudoheptulose and 7-phosphate and glyceraldehyde 3-phosphate. From pseudoheptulose, a block is transferred with the help of the enzyme transaldolase to here. So that fructose 6-phosphate is formed. So this is actually transferred to this molecule so as to become fructose 6-phosphate. So this involves a transfer type of enzyme again. And now the next step involves, so this is it, cellulose 5-phosphate, which actually is formed in one of the first reactions as a result of epimerization of Ribose 5-phosphate. So another molecule of cellulose 5-phosphate also comes into play here, as well as the erythrose 4-phosphate, which is taken from the just previous reaction. So these are the substrates here. We can see how it happens. So xylose 5-phosphate and erythrose 4-phosphate, which are having a combined carbon strength of 9. So that gives rise to, so out of those 9 molecules of carbon, 6 molecules are placed in fructose, and the rest 3 molecules goes to glyceraldehyde 3-phosphate. The enzyme involved is another transferase type of enzyme, which we have already seen in this step, transketolase. And what is the reaction? Yeah. So xylose 5-phosphate, a block of 2 carbons is taken from here and placed over here so as to become fructose 6-phosphate. And the remaining molecule is glyceraldehyde 3-phosphate. Actually, now the enzyme involved is a transferase class of enzyme, transketolase. So this is the last step of non-oxidative pathway or Hmp pathway, entire Hmp pathway, which we see now. This is non-oxidative pathway. The entire reactions are shown here, and you can see here, you can go through this. The carbon number can be traced here. So ribose 5-phosphate, then this combination, and this combination also we saw 6 plus 4, 6 carbon here. And one of the things you can see here to make the learning process more easier, it's always the phosphate group is always attached to the last carbon. As you can see here, the 7 carbon pseudoheptulose is having the last carbon, 7th carbon attached with the phosphate group. You take another case like fructose 6-phosphate, the 6th carbon, the last carbon. Here again xylose 5-phosphate, the last carbon, 5th carbon, and even glyceraldehyde 3-phosphate, that also the last carbon, the 3rd carbon. So this is another way. We have to always have to trace the carbon number here. So while we go through, while we learn this non-oxidative pathway, we have to trace the carbon number pathway, carbon numbers also, so as to remember this process more easily. This is the illustration. You can pause it. This is, we have already discussed non-oxidative pathway. And we move on to the regulation of Hmp shunt pathway. So whenever we go through metabolic reactions, we have to assess how they are being regulated also. Here in Hmp pathway, the first enzyme, this is the most critical, crucial, rate-limiting enzyme of Hmp pathway, glucose 6-phosphate dehydrogenase. It is inhibited by NADPH for obvious reasons, because we know that if the ratio of NADPH to NADP plus in a cell is very high, then this molecule can act as a sort of a feedback molecule so as to inhibit glucose 6-phosphate dehydrogenase. And insulin, one of our popular hormones, it is an anabolic hormone also. It actually enhances Hmp by inducing glucose 6-phosphate dehydrogenase. And Hmp pathway is more of an anabolic type of reaction and rather than catabolic type. Now, here we know that our cells are very economically conscious, energy conscious, and none of this metabolic processes are unnecessarily carried out. And here also the balance between glycolysis and pentose phosphate pathway, it depends on the need of the cell at that point of time, so that none of the futile, it will not lead to futile cycles. Then increase in fatty acid steroid synthesis enhances the pathway for obvious reasons, because fatty acids and steroid synthesis requires, and related related molecules require NADPH as a reducing potential. And not much significant in skeletal muscle, because skeletal muscle synthesizes very less of fatty acid or steroid molecules. So that's it. Thank you.